In this video, we're going to discuss the most probable number method. Step 1 is to make your 1 to 10 dilution. To do this, you're going to weigh out 10 grams of beef. You're then going to add 90 ml of buffered peptone water to the beef in the Whirlpack bag. Next, you're going to stomach that Whirlpack bag for 1 minute at 230 RPM. The next step is to create serial dilutions up to 10 to the minus 6. You're going to add 1 ml of each sample into the corresponding 9 ml buffered peptone water tube. Step 3 is to inoculate the mcconkey purple broth tubes. Timeout. Let's discuss mcconkey purple broth. mcconkey purple broth has a lovely purple color. It contains bromocresol purple, which is a pH indicator. Tubes containing acid-producing bacteria will turn yellow like tubes 2 and 3 in the picture. You may also notice gas production in the tubes. The arrow is pointing to Durham tubes. The tubes are made of glass and put into the tubes upside down. If the bacteria produce gas, you will see air bubbles in the Durham tube. Now back to the lab. We are going to inoculate the mcconkey broth tubes from 10 to the minus 4 to 10 to the minus 6. So first you're going to inoculate 3 mcconkey purple broth tubes with the 10 to the minus 4 dilution. You should repeat this for the 10 to the minus 5 and 10 to the minus 6 dilutions. Then we're going to incubate those tubes at 37 degrees Celsius for 24 hours. For step 4, we're going to check and record the mcconkey purple broth results. Check the color of the tubes. Remember that yellow equals acid production because of the pH indicator. Another positive indicator is gas formation. Remember that you can check for this from the air in the durum tubes. Step 5 is to subculture 1 ml from each positive mcconkey purple broth tubes into mcconkey purple broth tubes supplemented with mug. You should then incubate those tubes at 44.5 degrees Celsius for 24 hours. Step 6 is to check the results from the mac mug tubes. Tubes should contain acid and gas production. Here's an example of a positive mac mug tube. Positive mac mug tubes also fluoresce under UV light. The last step is to record the results from your mac mug tubes. Be sure to watch the video on reporting MPN results.